We're at the time where both antimicrobial resistance and neurodegenerative diseases are serious global issues. AMR is constantly increasing. We're running out of effective antibiotics, and we're looking for non-conventional methods how we could target antibiotic-resistant bacteria. And because antibiotics are failing, we really need to develop new methods and new approaches to target these infections. My name is Daniel Czyz. I am an assistant professor in the Department of Microbiology and Cell Science. Currently, my lab studies two different research areas. We study the role of gut bacteria on neurodegenerative diseases, such as Alzheimer's, Huntington's, Parkinson's, ALS, and so on. The other half of the lab studies non-traditional approaches to battle antibiotic-resistant bacteria. We employ bacteriophages, which are viruses that specifically infect and kill bacteria. And we also work on silver nanoparticles. We just recently found that a combination of silver nanoparticles with specific class of antibiotics, aminoglycosides, can potentiate the efficacy of these antibiotics by 20 times or more. That's very exciting given that some of these antibiotics are toxic, so we bring down the dose to a non-toxic concentration, but keep the efficacy the same or even improve. We also work on approaches, or what's called host-targeted approach, where we are targeting human cells to activate their ability to uptake and kill bacteria. Students are the brain, bread, and butter of the lab. My research involves both undergraduate and graduate students, and I really want them to think as scientists. In my lab, we
employ a lot of different methods. So first, we do a lot of in vitro work with bacteria. We're trying different ways to kill them, and we're employing different ways to assess killing. And that's usually through microscopy, spectroscopy. We employ the Caenorhabditis elegans, C. elegans model. And C. elegans are tiny nematodes. They have a short lifespan of about 3 weeks, so that gives us an advantage of doing, for example, lifespan experiments that we're also running. And they have a very simple body plan, so we could colonize them with any bacteria that we want. These worms are transparent, which allows us to monitor gene expression. If we tag any genes with a fluorescent marker, we could monitor the expression of these genes in real time without sacrificing the animal. So we do a lot of imaging. We also do mouse studies.
Within the field, we have been ignoring the role of bacteria on these diseases. But now that we recently found that bacteria can actually affect the pathogenesis and progression of these diseases, it opens new opportunities to treatment and management control. So we also recently identified first bacterial strain that can activate protective stress responses. And these stress responses can suppress toxic protein aggregation that is the culprit of all neurodegenerative diseases. So if we could use bacteria to activate these protective stress responses, perhaps we could alleviate the symptoms both at the molecular and organismal level.